This video will show you how to investigate the permeability of beetroot cells under different temperature conditions. First, we will use a cork borer to cut several long cylinders of beetroot. Make sure you use the cork borer in the middle as this is the widest part of the beetroot. Next, we want to cut the cylinders into the lengths that we require. First, we need to remove the skin from the ends of the cylinders. Leaving the skin on the beetroot would prevent the beetroot cells from releasing pigment into the solution due to its tough, thicker structure. Once the skin is removed, use a scalpel and ruler to carefully cut the cylinders into 1.2 cm lengths. You may find it useful to use forceps to hold the cylinder in place. Then, using the scalpel and ruler again, cut each cylinder into 6 equal discs. Next, we need to blot each disc with a square of paper towel to remove any pigment that has been released from the cells upon cutting. You should standardise your blotting method to ensure all discs are blotted evenly. This removes as much as possible the effects of any confounding variables in the method. Any excess surface pigment could diffuse into the solution and affect the results. Now we need to calibrate the colorimeter. We will use deionised water as our zero reference. Setting this zero reference is another way of standardising the method. Deionised water contains no pigment, so is often used as a blank solution to zero a colorimeter. First, set up the colorimeter with a blue-green 490 nanometers filter. Blue-green is the opposite colour to red on the colour wheel. This will allow us to measure the absorbance of a red solution, as the solution will absorb a blue-green light source. Fill the cuvette about 2/3 with deionised water and insert into the colorimeter. Make sure the transparent side of the cuvette is in the field of light. Press the R button to calibrate the colorimeter to 0. You do not need to touch the R button again. Label 3 boiling tubes with your initials and the temperature you will be investigating. There will be water baths set at different temperatures in the room. They are set to room temperature, 40 degrees, 60 degrees and 100 degrees Celsius. There will also be an ice bath. Using the syringe provided, add 5 cm cubes of pH 7 buffer to each boiling tube. Then cover each boiling tube with a square of foil. This will stop the buffer solution from evaporating in the water bath. Place your boiling tubes into the appropriate water bath. Allow them to sit in the water bath to allow the solution inside the boiling tubes to equilibrate. Once the solution has had time to reach the correct temperature, add 2 beetroot discs to each tube in the water baths. The discs need to stay in the water baths for exactly 10 minutes. You should use your stopwatch to time this. After 10 minutes, carefully remove the boiling tubes from the water bath. We now need to quickly sieve the beetroots out of the solution. Start with the first tube that you put the discs into. Collect the solution in a small beaker and return the solution to its boiling tube. Place it in a rack outside of the water bath and repeat with the other tubes. Make sure the discs have been removed before returning. Removing the discs ensures that the diffusion of pigment from the beetroot into the solution is halted. Once you have collected all your tubes of beetroot pigment, you can use the colorimeter to read the absorbance of the solution. Using the same cuvette you used to zero the colorimeter, 2/3 fill the cuvette with the solution from the first boiling tube. Place the cuvette in the colorimeter in the same way as before with the transparent side in the beam of light. To take an absorbance reading, press the T button. You will need to press the T button each time you want to take a reading. The absorbance value will appear on the screen. Record this in the table you prepared for your independent study. Empty your cuvette into the waste beaker and repeat. You will need 3 repeats for the 5 different temperatures.